this section of the notes is on the cell organelles, or the small parts inside the cells, their structure and their function. So the first thing that we want to do when we talk about cells is we really want to define what a cell is. So we've been talking about the cell membrane, and the cell membrane is actually a partition. It separates the inside of an area from the outside of an area. So every cell actually has a cell membrane. There are 4 things that all cells have. So cells have a cell membrane. We know that this is made of a phospholipid bilayer. It's really a mosaic of all these different components that work to make the cell membrane a selectively permeable partition. So we should already be very comfortable with what a cell membrane is. Well, the inside is what we call the cytoplasm, and this is the interior, and it's going to be mostly water. And so, of course, we know that this is going to influence what goes on in the cell based on whether things are hydrophobic or hydrophilic. But the idea that we just have this filling of the cell, it kind of acts like a cushion as well to provide the cell some structure and some support. All cells are going to have DNA. DNA, remember, is a nucleic acid. This nucleic acid actually has the directions to build proteins. And we've talked a lot about protein structure, the 4 levels of protein folding. We've talked a lot about protein function, what proteins can do inside cells, in the membrane. So we've talked a lot about proteins. We know that proteins are really what does the work in the cell. And DNA has the directions to build the proteins. We do want to keep in mind that we're going to kind of build these selectively. When we need them, we'll build them. If we don't need them, we might stop building them. And so in order to build a protein, we actually need a part of a cell called a ribosome. We've actually talked a little bit about the ribosome already. We know that this is rRNA and protein, but this is where proteins are built. So every cell will always have these 4 things. How much, how little, the way that they're differentiated, that's what makes different cells different. So one of the things that we'll look at in this unit is the idea of unity and diversity. So what do all cells have in common? How are different cells different? And of course, you're used to talking about prokaryotic and eukaryotic cells, but we're also going to be talking about the idea of differentiation even in eukaryotic cells. So we can have what are called undifferentiated, and stem cells are an example of undifferentiated cells. And as they more and more selectively use their DNA, they become differentiated. So we'll have cells that become part of the pancreas, cells that become part of the spinal cord and the brain, part of the heart, part of the bone, part of cartilage. And so we're really going to be looking in this unit at how are all cells similar, and then how do cells use their internal structures to become more and more specialized. So the very first thing that we actually look at are the 2 main types of cells, which are prokaryotic and eukaryotic cells. So we just talked about what all cells have in common. The main thing that we really want to focus on is this idea that DNA is what we call universal. All cells are going to have DNA. They'll use it a little bit differently, but they all have double-stranded nucleic acids. It's all made of nucleotides. It's all made of A's, T's, G's, and C's. And so it's just how that's used that makes the cells different. One big difference that we're actually going to see in prokaryotic and eukaryotic cells is, of course, the idea of where is the DNA. In prokaryotic cells, the DNA is going to be in one circular chromosome. It's still a large chromosome. It's still going to coil and fold. It's going to be found in a region called a nucleoid region, but there is no membrane surrounding. So our two main types of cells, we differentiate between the first dividing of cells, the first classification of cells, based on whether they have a membrane surrounding. So no membrane surrounding are known as prokaryotic. And kary actually means nucleus, and pro means before. So if you look at the actual word prokaryotic, you can break it down into what the word means before a nucleus. No membrane surrounding the DNA. The advantage to this is it's easy to get to the DNA. The disadvantage is it's not very well protected. It's not very well organized. If we look at eukaryotic cells, eukaryotic, the prefix EU, means true. So they have a true nucleus. All that a nucleus is really a membrane surrounding the DNA. The advantage is we're able to protect the DNA, we're able to partition. Anytime we have a membrane, we talk about it being kind of a partition. We're able to partition it off so that it's separate from the rest of the cell. Inside of the nucleus, we're going to have what's called a nucleolus region, which we'll talk about as well. The main idea being that eukaryotic cells are actually going to, therefore, because they can protect the DNA, their DNA is going to be larger in amount, so a larger amount, and it's going to be more complexly packaged. The other thing that we see about eukaryotic cells, besides the fact that they have more DNA and more complex DNA, is they're actually going to have more complex structures. These are going to be our membrane-bound or membrane-surrounded organelles, things like the endoplasmic reticulum and the Golgi. All cells have some type of membrane. It just depends on the amount of membrane that's being used and what those membranes are being used for. So when we look at what all cells have in common, we're kind of trying to keep in mind that they have four main things in common with each other. One of the most important things being that DNA is universal. Every cell has DNA. Then we start to look at how they're a little bit different, so comparing and contrasting the cells. We do want to keep in mind that prokaryotic cells are usually smaller, and they're called simpler. But we're going to talk about how prokaryotic cells are kind of like a moped compared to the Ferrari of eukaryotic cells. They still get you somewhere. They still do the job. They're still a cell. They just have different ways of performing the functions. So one might have a V8 and one might have a V4, that sort of idea. They still get the job done. So what we want to focus on right now is we actually want to focus on prokaryotic cells and what structures do they have in common. And so when we look at prokaryotic cells, prokaryotic and bacteria basically mean the same thing. Prokaryotic cells are divided into two types of bacteria. We're going to have eubacteria, not the same thing as eukaryotic, Eubacteria just means true bacteria. And we're going to have Archaea bacteria. They have a lot in common. They also have a lot of differences. We'll talk about the differences more later in the year when we do our kingdom survey. But the big idea is that they're both prokaryotic. They do not have a nucleus. No membrane-bound organelles. So we want to look at what structures do they have. They don't have a nucleus, but what do they have? And how do these structures help the bacteria function? So if we look, we can see some of the things that we talked about before. We can see where we talk about nuclear material. This just means DNA. So we see DNA, we see the cell membrane, we see the cytoplasm, and the ribosomes are here, they're just not labeled. So all these little dots, we're going to go ahead and label the dots ribosomes. The structure of a ribosome is actually much more complex. You've already seen a picture of this, but we are usually going to show them in a picture as just dots. What we notice is that there are other things than just those four. One of the main ones we want to pay attention to is the cell wall. The cell wall, we already know, is made of peptidoglycan, which is a combination of peptides, proteins, and glyc sugars. So the cell wall is made of sugars. It's there to provide support. Right, we know that the cell wall is going to provide some pressure back in on the cell. It's going to really support the cell. We do actually see a region where the DNA is concentrated, but you can see it kind of takes up most of the area of the cell. Glycogen granules, we're not going to really worry about these. In fact, we're going to cross them off. We do want to focus on what's called extrachromosomal material. So I need you to draw kind of a little circle, and we're going to call this a plasmid. P-L-A-S-M-I-D, plasmid. A plasmid is going to be what we call extra or beyond the chromosomal material. So the DNA in a bacteria is a single circular chromosome, but we do have small plasmids, which are extra pieces of DNA, and it turns out, they can be traded. So we'll talk about these more when we get into biotechnology and the use of biogenetically engineering bacteria. For now, we do want to know that they do have some unique features. They're not just the most basic cell. They have some components that help them trade information. This increases variety. They can also have something called a capsule. You think of what a capsule is when you're taking like a pill and there's a capsule around it. It kind of surrounds it a little bit more. The capsule is going to be the most external surrounding. And this actually helps it stick to other cells. So one of the things that we'll talk about in regards to bacteria is that they can be pathogenic. They can stick to our cells and cause disease. And so this capsule and these stringy things, which are called cilia, this yellow capsule is going to help it actually stick to your cell. So a really good example are the bacteria that cause intestinal distress or food infections versus food poisoning. And these will actually stick to the cells of your intestine, actually release a toxin that makes you really sick. The last thing that we want to talk about is the flagellum. And the flagellum of a bacteria is a single protein that is able to rotate, and this is able to provide movement. So bacteria actually are able to move, some of them. So some of them have a flagella, and that will help them be able to move. So what we see when we look at bacteria is that we have some really simple aspects and some really complex aspects, all of which help the cell function.